for most psychiatric disorders and most people who have a psychiatric condition, their form of it, their genetic risk will be in that form of polygenicity, that is, additive effect of small variants, none of which individually are sufficient to cause a disease, but adding hundreds of them up gives you sufficient risk. And when we look at that polygenic risk, we can kind of make a score out of that, out of adding it up. And we can compare the scores from different disorders, and they do overlap kind of remarkably. Not 100%. Let's say autism, polygenic risk, and schizophrenia might correlate with each other about 20%. And so that tells you that most of the risk is distinct, but that there is some shared risk.
Right.
In fact, we've done studies with collaborators in Denmark, and Denmark's a really interesting country because they have genotyped in a project called iPsych pretty much every patient who's gotten a hospital-based psychiatric diagnosis over about a 15-year period. So if you think of that, it's 80 or 90,000 people and then another 20, 30,000 controls. Autism, schizophrenia, bipolar, major depression, eating disorders. And we decided, instead of treating them separately, let's just call anybody who has one of those disorders as somebody affected and somebody who doesn't have one unaffected, and do a genome-wide association study. So there we're not looking for genes or loci associated with autism, schizophrenia, bipolar. We're looking for things that are just generally associated with having any of these mental illnesses. And lo and behold, one finds dozens of loci that aren't related to risk for an individual disorder, but that are related to risk for almost all of these disorders. And those studies have been expanded, and now there are over 200 loci identified that predispose in this kind of additive polygenic way to just having a mental illness, unspecified.
Right.
Which is kind of wild. It just tells us that maybe mental illness is just kind of one part of our normal, of normal human variation, and that there are a series of genetic variants that make people more or less resilient to the things that happen to us in our lives that make us depressed, that make us anxious, that might make us have an eating disorder, etc.
Dr. Geschwind, thank you so much for joining the UCLA Brain Sport podcast. Let me intro you so that everyone knows who you are. You are the Gordon and Virginia MacDonald Distinguished Professor of Neurology, Psychiatry, and Human Genetics at the UCLA School of Medicine, and you are the Senior Associate Dean and Associate Vice Chancellor of Precision Medicine at UCLA. So I've heard you say that we are in a revolution of healthcare. We're also in a revolution of what we know in the field of neurogenetics.
Absolutely, yes.
Can you maybe extrapolate a little bit more on that? Because that sounds very exciting.
Yeah, no, it would be great. Now, first, thanks for the opportunity to talk about my work.
Thanks for coming. Yeah,
because it's really fun. So, two things have happened. One is amazing increase in computer power.
Right.
And it's called Moore's Law, is one of the things that predicted it. And so Gordon Moore in the mid to late 60s said, I think it was like 65, 66, said, hey, by the way, you think things are amazing now, making these integrated circuits, these chips. In every year, the density of transistors on them, of components, is going to double over the next 10 years. So think about that, right? So this exponential increase in computing power that's going to occur. Well, he was wrong, and he changed his whole law to be it's going to double every 2 years, but it's been doubling every 2 years for the last 50 years.
Right.
And so that's an incredible change in computer power. So that's one revolution that's occurred. The other is sequencing. So our ability to sequence the genome went from $3 billion per genome, 10, 15 years ago to now closing in on $100. That is an exponentially faster growth than what happened with transistors and integrated circuits.
Right.
So you put those two things together, the ability to produce huge data sets, because the genome is 3 billion base pairs, each person, so it's a lot of data, and the ability to compute it and to store it and to use it, and our burgeoning understanding of what it means, and you really have a revolution. The ability to use genetic data to understand individual susceptibility to disease, their resilience, their risks. So we're in a whole new era. Yeah, so it's those two things, the increased computer power and the massively increased ability to read our genomes, store them, and interpret them. That's really been incredible. And it's not just reading the DNA sequence, it's using the same kind of sequencing power to look at things like epigenetics, which we can discuss, as well as other genomic features like RNA expression, et cetera. And so, we have all that information, but the precision health also can utilize all kinds of non-genetic information, right? We can use just big data in general, like the health records, and look at, if we have enough people who've experienced similar things, we can begin to see, can we predict if an adverse outcome is going to happen? We can use the genetic data, but it's not dependent on that. So for example, it may be that 4 or 5 different clinical markers can tell you, and you don't need the genetics. So,
Right.
So there are all different kinds of ways that precision health is using big data. One of them might be, we send a lot of people home from the emergency room. We want to prevent them from coming back. Maybe we can predict before we send people home who is at really high risk for coming back within 24 hours and being admitted to the hospital to avoid that. It's bad for the patient, it's bad for the doctor, it's bad for the system. And similarly, we might send patients home a little too early, especially elderly patients who might be at fall risk. So can we predict who's at very high fall risk and take measures to remediate that before they go home, or can we monitor them, et cetera? So there are all different kinds of ways that we can use big data, electronic monitoring, as well as genomics. But genomics provides an interesting new way to understand our individual differences from each other. And that's really where the personalized medicine piece of it really comes in.
Right. So you mentioned Moore. Did he predict machine learning into that exponential growth in our capability? And if not, and if so, what has the contribution of machine learning been to our capability?
Well, I'll frame it a slightly different way, hopefully try to answer that, which is that as early on, the data wasn't that big. I mean, what I carry in my pocket now, this little phone, is equivalent to a room full of IBM computers when I was a kid.
Right.
It's more powerful than anything like that. So I would say for that first decade or two, there wasn't that much data. It didn't really necessarily need machine learning. In fact, it's amazing what we've been able to do with relatively simple computers and calculations, including putting a man on the moon, people on the moon. So, but as data gets bigger and bigger, it gets harder and harder to handle, to analyze, and one can use machine learning and artificial intelligence and different forms of deep learning to kind of tackle problems that may be too many dimensions for the human mind to really grapple with. But of course, there are risks there as well, which we can discuss in more detail. And I'm not talking about the risks of the machines taking over the world. I'm talking about very simple risks, like if you tell a machine to find the difference between two, say there are cases and controls in these data, they'll find it. the question is, does it mean anything? And so it's really critical to do the machine learning properly, to make sure that there aren't latent variables that are in the data that actually would make two things seem different when they're really not, when it's a confounding variable, like age or what somebody ate for breakfast and things like that, that could influence what you measure later in the day.
But don't you think that inherently, like, machine learning involves the machine learning, right? So the more that you use it to be able to identify those associations and then telling it, there is no association here because of this confounding or that confounding or this variable.
It can learn.
It learns, right?
No, absolutely. So and so what you just brought up is a really key point. I think the essential thing is that you have it learn, and then you actually test an independent data set how good it is at predicting what you've asked it to do.
Right.
And it can keep iterating on that and keep improving.
Yeah.
But what's been happening a lot is that people run a machine learning algorithm on a data set and then say they've found a way to separate Parkinson's disease from controls, when really what they've done is they've just found some confounding factor in the data that separates them to some degree. And so when you apply that on an out-of-the-box sample, it actually doesn't work that well.
Right.
So that's something you just have to be careful about. But what you mentioned is the way it should be done.
I think there's, and you've mentioned this before, that, there's also an influence of not enough big samples, right? And not enough enough genomes mapped, right? So the more genomes that you map, the more significant you find those differences that, in a lower sample, it might just be an aberrant association, but the more samples that you get, the sure that you can be that there is actually significance in those differences.
Right, and it has to do with the fact that the data has very high dimensions, and so that when you take a genome from one individual, you're testing a lot of hypotheses there.
Yeah.
There are a lot of different variants, millions of variants in that individual's genome. And so to actually identify a variant or several variants that would differentiate those at risk for disease or risk for an adverse outcome from those that don't have that, you need a lot of people to have statistical power because of the multiple comparisons issue. You're making a lot of comparisons, and you're going to find a lot of things by chance if you don't have a large enough sample and you aren't rigorous enough about the correction.
Right. And you mentioned that mapping a genome used to cost, $1 billion or something like that before, versus now it just costs maybe $100. What about the amount of time that it takes to map an entire genome, several years ago versus now?
Right, now it's done in hours or a day. There are two parts to it. One is sequencing the genome, and when I was talking about the $3 billion, that was actually making the first real sequence map of the genome, but it was only about 60% complete. And even now, our knowledge of the genome isn't fully complete, but it's close, much closer. And so now what we do is I sequence your DNA, your genome, and I map it to the genome to see, to read it. It makes it a lot easier than assembling it from scratch. So it has like a backbone, we map it, we read it, and that process can be done, depending on what kind of computer you have, kind of in a few hours now for a single genome.
Yeah. So let's start talking about, because we talk about this all day, right? But let's start talking about specific diseases. You've done a lot of research with autism spectrum disorder and other neuropsychiatric disorders, which is a wide spectrum, right? You have anxiety, PTSD, bipolar, major depression, all sorts of stuff like that. Are you able to provide a genetic basis for those diseases, like in general?
Well, it depends what you mean by genetic basis, but let's talk about autism for a second. Let's say 30 years ago, it was known that there was a large amount of heritability. That means 70, 80%. And we knew that because when you looked at monozygotic twins and you compared them to dizygotic twins, that is, identical versus fraternal, that people who shared 100% of their genome, the identical twins, had almost 100% concordance for autism. That is, if one were autistic, the next would be too.
Right.
The dizygotic twins, it was maybe 1 in 5, 1 in 10.
Right. Can you describe the difference between those two kind of twins for everybody?
Yeah, the identical twins, 100% of the genome is the same, and the fraternal twin or dizygotic twin is like a sibling, so 50%.
Okay.
So when you see the drop-off in sharing of a particular phenotype, which is anything we can measure, like autism, depending on the degree of relatedness, you can calculate the heritability based on that.
Okay.
So when you have 100% of your genome shared, you have nearly 100% concordance. That is, if one twin has the disorder, the next one is almost certain to.
Okay.
Whereas if they're fraternal twins, it might be 1 in 10 or 1 in 20
Okay.
or 1 in 5. Somewhere like 10 to 20% of the second twin will share the autism. Now, and so so we kind of knew that, but we didn't have many genes. We didn't know what genes it was. We didn't know how heterogeneous it is. But what we've discovered is that autism is highly heritable, about 80% general heritability, and that there's a component of autism, maybe up to a fifth, that is more like Down syndrome. It's not heritable, but it's genetic, so that you have mutations actually in the germ cell, in the egg or the sperm, that aren't in the parents, but that occur maybe due to paternal age or other factors, and that causes autism. Those are called de novo or new mutations, and that may be 10 or 20% of autism. And what we found there is literally around 200 plus or minus genes that we've identified that we have pretty high confidence cause autism by this mechanism, in that they are de novo mutations that aren't inherited from the parents, and but that occur in the child with autism and are sufficient to cause the autism. And often with those mutations, you'll see other comorbidities like intellectual disability or motor dysfunction or seizures or language delay as well. Now, that means that that component of autism is a collection of rare disorders. So you have about 10 to 20% of autism that's a collection of rare disorders, including things like Fragile X syndrome. About 20% of kids with Fragile X syndrome have an autism diagnosis.
Yeah. Well, I think that also, and something you mentioned before, is that a lot of these diagnoses are relatively subjective, right? Like, we don't have any objective marker to say, if this is high or if this is present, then yes, you do have autism. Of course, something that's not genetic, right? It's not like a biomarker or something like that.
Yeah, I mean, the autism is a clinical diagnosis based on history and examination of the patient. There is not one single thing. It's not like tuberculosis, where you can actually measure the bacteria and say, aha, this is tuberculosis pneumonia.
Right.
And so, but what's interesting about psychiatric diseases is, despite the kind of fuzziness with which the diagnoses are made, there is some underlying truth in the neurobiology, because we can use these clinical diagnoses, collect large samples of patients, and if we do it properly, we find a coherent association signal, a genetic signal. And so even though there is some haziness around the diagnosis and fuzziness, it's good enough to identify disorders that are biologically coherent. The other way that we know that we're on the right track to some degree is that when you take something like ADHD or major depression, we have medicines that work. So if they have the diagnosis and I give somebody with depression a certain drug that's for depression, it has highly, it's going to work. I wouldn't give that drug for schizophrenia. It's not going to work.
Right.
And for ADHD especially, the stimulants are highly, highly effective.
Yeah.
And so again, I think there is some biological coherence. Now, that doesn't mean that these are one disorder. It means that they're syndromes with many, many causes, and that as we gain more and more genetic knowledge, it will allow us to maybe subset and refine our biological understanding. So I'm not saying that we have a great biological understanding of these disorders, but I'm saying that it's kind of remarkable, despite how fuzzy these diagnoses are. Like I compare it to something like Alzheimer's disease or Parkinson's disease in neurology, where we can get a definitive diagnosis by looking at the brain post-mortem.
Right.
There is no post-mortem pathology that we can look at in these disorders. So they're defined by observation of behavior and cognition. And despite that, there still is some coherence there in terms of defining what these disorders are. And that's been kind of surprising.
Yeah, but there's a lot of overlap in terms of a lot of the symptoms, especially for the psychiatric diagnoses, right? Like,
well, that's really interesting. That's a really good point. So some of them overlap at the symptom level. There are some overlaps. And there are many people who carry multiple diagnoses as well. And so maybe it's not surprising then, when we look at what we found genetically, we can find overlap between disorders. Things like autism and schizophrenia, which clinically are really quite different when you think about their natural history, autism occurring early and being observable early, and and having really quite different phenomenology than schizophrenia. And yet there are some rare genetic conditions, known mutations, that can cause both disorders.
right.
Things like 22q deletion, DiGeorge syndrome, can predispose to autism or schizophrenia. And we see this with a number of genetic variants. And then if we look at common genetic risk, which is in the people who don't have a major effect size, big mutation like Down syndrome or Fragile X or something like that, but we're just looking at the additive effect of many genetic variants, which is called polygenic risk.
Right.
And for most psychiatric disorders and most people who have a psychiatric condition, their form of it, their genetic risk will be in that form of polygenicity, that is, additive effect of small variants, none of which individually are sufficient to cause a disease, but adding hundreds of them up gives you sufficient risk. And when we look at that polygenic risk, we can kind of make a score out of that, out of adding it up. And we can compare the scores from different disorders, and they do overlap kind of remarkably. Not 100%. Let's say autism, polygenic risk, and schizophrenia might correlate with each other about 20%. And so that tells you that most of the risk is distinct, but that there is some shared risk.
Right.
In fact, we've done studies with collaborators in Denmark, and Denmark's a really interesting country because they have genotyped in a project called iPsych pretty much every patient who's gotten a hospital-based psychiatric diagnosis over about a 15-year period. So if you think of that, it's 80 or 90,000 people and then another 20, 30,000 controls. Autism, schizophrenia, bipolar, major depression, eating disorders. And we decided, instead of treating them separately, let's just call anybody who has one of those disorders as somebody affected and somebody who doesn't have one unaffected, and do a genome-wide association study. So there we're not looking for genes or loci associated with autism, schizophrenia, bipolar. We're looking for things that are just generally associated with having any of these mental illnesses. And lo and behold, one finds dozens of loci that aren't related to risk for an individual disorder, but that are related to risk for almost all of these disorders. And those studies have been expanded, and now there are over 200 loci identified that predispose in this kind of additive polygenic way to just having a mental illness. unspecified.
Right.
Which is kind of wild. It just tells us that maybe mental illness is just kind of one part of our normal, of normal human variation, and that there are a series of genetic variants that make people more or less resilient to the things that happen to us in our lives that make us depressed, that make us anxious, that might make us have an eating disorder, etc.
But there's some psychiatric diagnoses that are distinct and much more severe than others, right? Like you talk about schizophrenia versus anxiety, right? I do wonder, is there are some psychiatric disorders more polygenic versus other psychiatric disorders like schizophrenia where you're hallucinating and it's significantly debilitating? Do those have less polygenic but more one sort of genetic mutation or genetic profile?
That's an interesting question. It's actually maybe a little counterintuitive, but schizophrenia is vastly polygenic. The vast majority of its risk is polygenic. Studies have found rare major effect variants in some cases, but it's far less than in something early onset like autism. So think of it another way, is maybe it has to do with the onset. So that something like autism, which occurs very, very early, requires these kind of larger effect mutations, or they're more likely to be having a role, whereas something like schizophrenia or depression is more likely to be polygenic. Now, that being said, still, the majority of autism risk is likely to be polygenic.
Right.
But there's much more rare large effect mutations in autism than in schizophrenia. Now, if you compare schizophrenia to major depression, certainly there aren't any large effect mutations that have been found that cause major depression. So major depression is more polygenic and more of a gene-environment interaction than, let's say, schizophrenia. That's for sure. The other thing I wanted to say about anxiety is that from my perspective as a neurologist, from a biomedical perspective, I think of anxiety as a symptom. It can be a disorder in some people. I think that's rare. Most of us feel anxiety sometime or another. If I make you hypoxic or you make me hypoxic, I will get anxiety.
Right.
And so often in the hospital, anxiety is a symptom of something else. And anxiety, therefore, travels along with a lot of different neuropsychiatric disorders as well as other biomedical disorders.
Let's go down a little rabbit hole. Do you think anxiety is pretty common? It's one of the most common psychiatric issues in our society.
Yes.
Do you think that might be the case because anxiety evolutionarily can be beneficial?
Yes.
Just yes.
Yeah, one of the questions we have to ask is, polygenic risk is polygenic because each variant has very, very small effect size with regard to the condition that you're saying it causes, like autism or anxiety. So that means it takes hundreds or thousands of these to add up to have that. Why is that? It's because they've been under natural selection for thousands of years, tens of thousands of years, and selected out of the, large mutations that have large effects are selected out if they cause deleterious phenotypes.
Right.
And so one has to think that maybe natural selection has done a lot to take away things that are, let's call them bad, and left things that are adaptive in the population. Now, that's an interesting thought. What we know about, let's say, metabolism. So why do I have to take cholesterol medication? Well, it's because my body is adapted evolutionarily, probably 1,000 years ago, my ancestors, to have high cholesterol, because that was good. because I probably only had a meat meal once or twice a month, maybe, right? And I had to preserve every single calorie that I ate and, to live. Now, when there's food, I can just snap my fingers or go on my iPhone and order whatever I want to my door from DoorDash.
Right.
That's kind of changed things. So the environment has changed faster than our genomes have changed.
Yeah.
So we have to be careful about how we interpret the genetic variation that we see in the context of the current environment.
Right.
So getting back to ADHD, for example, right? ADHD, like, the things that cause ADHD, all of that genetic variation, where it's very common, were probably super helpful. And until children had to sit in school for 8 hours a day, which is only in the last 100 years, and in most populations, the last 50 or 60 years, where most children are in school, that's a diagnosis that comes out of our social situation, right? We've created it by creating school.
Right.
That would be one way of thinking about it. Now, preschool, like before we had school, ADHD might have been really useful in that somebody was able to multitask and pay attention to things, and they would only pay attention to the things that were really important and didn't want to listen to somebody droning on and on and on about some mathematical something or something they weren't that interested in.
Right.
So let's get back to anxiety. Anxiety could be, general anxiety, where somebody's thinking about, worried about things happening, makes people vigilant. And if you live in a society where there are a lot of animals that can eat you, you should be vigilant.
Yeah.
And most people don't live in that. But actually, if you walk around in Africa in the bush, you become part of the food chain very quickly. But most of us have never experienced that.
Right.
So that kind of makes it pretty clear that it's probably smart to be pretty vigilant and to stay out of the bush. And if you are in the bush, carry a spear, stay clear of elephants, etc., all those things. And thing with snakes. Some people are scared of certain insects or snakes or other things, and it's probably because those things carry disease and or can hurt you.
Right.
And so that's probably pretty adaptive.
Yeah.
Sorry for going on and on about
It's
that.
okay. It's okay, because this leads in perfectly into something that you said that I want to quote back to you. And it's a really, really interesting concept. And I don't care what else we cover on this podcast, as long as we can cover this. This is probably one of the more interesting parts of the podcast, in my opinion. You've said that aspects of psychiatric disease risk are related to human cortical expansion and human brain evolution. You've also said that genetic regulation drives human brain evolution through the expansion of our cerebral cortex.
Yes.
So I got to ask, do you think that this brain expansion secondary to the genetic regulation has now predisposed us to more psychiatric illness and more psychiatric disease?
Yes, but I think we have to unpack that.
Okay.
Yeah, which I'm glad you want to do. So what's happened over evolution is that the mammalian brain has, all these amazing adaptations that have occurred. And now you want to kind of build and continue to grow the brain and grow its connectivity and what it can do. And a computer is a bad and inaccurate analogy, but kind of the more processors you have, the more things that are connected, the more processing it's going to be able to do. And so as you kind of get more and more of that online, how do you do that when you already have something that works? And so it turns out that all of this technology and all of our genome advances have taught us several things. And I'm going to divide it into two areas. One is the genetics of disease, like what predisposes to human disease. And the other is understanding what makes us different from other organisms. And so we can sequence the genome, we can sequence RNA, we can look at what's expressed, and what we find is that brain genes are under what's called much more negative selection or purifying selection than non-brain genes. And that means that there are many fewer mutations or variants in brain genes compared with other non-brain genes or ubiquitous genes than you might expect. So in other words, brain genes can't tolerate a lot of difference. So then how do you reorganize? How do you adapt? How do you begin to build more if that's the case, if brain genes are so sensitive to dosage and other things? Well, you change their regulation. You change when they're turned on and off. You change how they're used. You might change how they're spliced together. You might put in slightly new kind of components in the gene, like something might get spliced in that wasn't present before, or something might get spliced out, which changes its function a little bit. So it's kind of like tweaking.
Mm-hmm.
And so most genetic variation sits outside actually of genes, and certainly outside of the protein coding part of genes. It sits in regions that are likely to be regulatory. So only 4% of the genome codes for protein. So when we begin to do these genome-wide association studies for diseases and all kinds of human conditions or human phenotypes like height, weight, and things like that, we find that most of the genetic variation is not sitting in the protein, that 4% of genes. Almost 90% of it is sitting in regulatory regions.
Mm-hmm.
And so that's pretty interesting. Let's put that in our back pocket for a second. So
Sure.
We kind of know that. So then the question is, well, we know that brain genes are under this constraint, or under purifying selection. So the protein coding part of those genes doesn't change very much. It means they can't tolerate much difference from person to person. And if you do put a mutation in them, it's likely to be pretty deleterious. However, the regions that control the expression of those genes, when they're turned on, what cells they're in, as well as their splicing and other things, those regions do change and can change, and they're not under the same level of constraint as the protein coding part. And that's really what's happened. And it's actually very interesting. The first paper that really came out to kind of state this was a very famous paper in 1976 in the journal Science by King and Wilson, where they sequenced proteins from, and not too many, really just a few proteins, based on a few protein sequences between humans and other species, where say, well, there's very little difference between humans and monkeys.
Mm.
Therefore, it can't be the protein coding part of the genome that's changing.
It's got to be the regulation.
It has to be the regulation. And so now we actually know that, and we can measure it. We can see that changes have occurred in regulatory regions. So then the question is, where are these changes? What kind of genes are they affecting? And so we've been able to use work that other people have done where they've identified regions that change in humans more than you'd expect by chance, called human accelerated regions. And these are usually things called enhancers that that regulate gene expression. They are the non-coding regulatory part of the genome.
Mm-hmm.
So they would be switches that help refine how high a gene is expressed, what cell it's expressed in, what time it's expressed, that kind of thing. And so, and we can find that many of those things are connected. The things that are changing the most, these human accelerated regions or other things that are changing more in humans than you expect by chance, those are regions that control genes involved in brain development more than you'd expect by chance.
Mm-hmm.
So it's telling us that a lot of what's happened in evolution are controlling genes that affect brain development. Then we can ask, Okay, how about the genetic risk for disorders that involve brain development, like autism or other things? And when we look at those, schizophrenia, they do impact more than you'd expect by chance these human accelerated regions or other regions like that. And they impact the mutations that I told you about earlier, that these de novo mutations in humans that contribute to autism. Those genes are genes that are also more likely to be controlled by human adapted elements.
Mm-hmm. What about the non-de novo ones? So meaning the polygenic ones, the ones that are additive, the ones that are common variants in our society?
It's a little bit less clear. It's it's not quite as strong, but there is evidence that. So let's think about it this way: genes, even the de novo genes, but even the polygenic genes don't cause disease. I mean, we didn't evolve to have autism. We didn't evolve to have schizophrenia. We didn't evolve to have severe anxiety that's debilitating. So all of these things, all the polygenic risk is related to regulating things that are probably adaptive in the right dosage. It's just that if they get mixed the wrong way, you can have a little too much of a good thing, let's say, and it can be maladaptive in our current society, the way that our current society works.
So you would say, from a genetic standpoint, and this is a reductionist view, but these psychiatric issues and maybe autism is a byproduct of where we are now in our genetic profile, secondary to natural selection.
Yeah, and it's but they're adaptive, because natural selection is only going to let through things, it will let through things that are neutral or adaptive. It's going to weed out things that are maladaptive. And so I'd even go further than that.
It would weed out things like huge mutations that would be maladaptive and not allow you to survive and pass down.
Sure, it would weed out that. And then even in a smaller way, things that can work in an additive way to be bad for you, it would also weed that out. The issue is that it doesn't act as quickly as our society changes. So I gave you the example of metabolism.
Yeah, great point.
It would be nice if I could just adapt, so I wouldn't need a Lipitor. Right? If my body could just adapt to use less nutrients and my appetite could go down a little bit, et cetera, right? And so, but I want to make another point about that. So do we have evidence that the polygenic risk for things like autism might be adaptive? Well, one of the pieces of evidence is that if we look at the polygenic contributions to educational attainment, or IQ, educational attainment means how many years of schooling you had.
Yeah.
IQ, related to IQ tests. And those two things are highly correlated with each other. Usually the higher the IQ, the more educational attainment on average. But of course it's not a one-to-one thing, because how much education you get is also part of society, where you grow up, what opportunities you have, et cetera. So if you look at autism polygenic risk and we ask, how does it correlate with other brain features, brain phenotypes, like educational attainment or IQ? It shows a positive correlation. That means that there's about a 0.25 to 0.4 correlation between risk for autism and the risk to have high IQ or high educational attainment.
Right.
So these are things that are, that we think of as adaptive, as positive in our current society. So it is pretty interesting that, and the key thing will be now to understand, what's that neurobiology? What does lead to high educational attainment or high IQ? What are these polygenic risks or polygenic factors leading to, right? Like, what are they changing? Are they changing in a very subtle way the way synapses work, or the way glial cells work, or whatever, right? Where does that sit? And that's a really interesting question. And I think genetics will help us answer in the next decade.
Yeah, and I think the clinical phenotype of, your archetypal, autistic person that is a savant in,
Some area,
some
yeah.
other area.
Well, yeah, sometimes there's one characteristic is high focus on things, right?
Yeah.
Like a lot of focus.
Right.
And that can be very beneficial. But in general, what this brings us to is thinking about a lot of what we think of as disorders today are just part of normal human variation. If we had a society where we had to be able to draw really well rather than write really well, I wouldn't have a job, right? And so these are just things that are part of normal human variation. And what we call a deficit is sometimes a byproduct of the society that we've created and what it takes to be a quote-unquote successful person in that society. And it really feeds into the neurodiversity movement, which has really made these points much more forcefully than I'll be able to make them, which is that this is really about individual differences in the vast majority of cases, and we have to recognize that there's a wide range of individual capabilities, and that it's our society that makes it hard, easy, adaptive, maladaptive for many of these things. And so we have to think about what kind of society we want to have.
Great point.
Shouldn't we be accommodating people with disabilities,
Right.
Which we call disabilities, but at the other level, it's a difference. So I'll leave it at that.
Yeah. And we could go down that rabbit hole, and I'm going to circle back to that when we talk about maybe some environmental factors. But and this might be a stupid question, but do you think that our brain is still evolving and still growing and still undergoing, natural selection to be able to, continue to expand our cortex?
Well, that's a great question. Expanding the cortex is, so our cortex has expanded, as of other things, but our connectivity has expanded massively compared to our next closest relatives, like in the great ape and, chimpanzees. But within humans, there's no relationship other than, if you have microcephaly or, between brain size and kind of functioning, right? Like within the normal range. And so it's not as much the brain size that I would say that's occurring right now, but, so brain size may change over millions of years.
Right.
Right. I mean, like we're talking about the difference between us and our closest relative is 4 to 5 million years
Right.
of evolution. So certainly over that time frame, should we not screw up things badly enough and still be around, that there's likely to be other species that have emanated from us that hopefully have better brains, or they may not. They may evolve because of changes in the environment that make them more resistant to certain kind of plagues or heat or changes in temperature. Who knows? But the kind of shorter term evolution that would come, you evolve to, you adapt to your habitats. And so, you can imagine as if we become a more and more sedentary, more computer, more connected society, that certain kinds of brain functioning is going to be more adaptive than others.
I'm just trying to tie in whether you would anticipate, and it's a tough question, right? But whether you would anticipate that evolutionarily you would foresee that there will be more psychiatric diagnosis in the future, right? Because we're seeing an increase in psychiatric diagnosis, and certainly that can be secondary to all these other environmental factors. But I'm wondering if you foresee that in the future, or if you see new psychiatric diagnoses coming about in the future, secondary to it's that same concept.
Yeah, it's interesting. I don't think the brain has that many ways of responding. So I think what we see and what we've diseasified or called disorders that has to do with maladapting to society isn't going to change that much. Of course, the DSM has changed, massively,
Right.
since it's been developed, but that's not a real change, right? That's just a change in how we categorize things.
Right.
So certainly how we categorize people will change over time. But I don't think our brains, like, for example, neurodegeneration that occurs with dementias is likely to be a lot of final common pathways working their way through. Our brain doesn't have a lot of ways to adapt. And so I think that a lot of the alteration, a lot of the variability in behavior that we have just reflects the human condition. And so will we have more variability? So much of that depends upon what the selection pressures are and what happens. So there's, I don't really have an answer to that.
Yeah.
Certainly not anything that I've thought through and that's intelligent.
Yeah.
What I would say, I think just the important message to take home is that there's a huge variation in human abilities. And often, things that in our current society look like they may be weaknesses from a brain standpoint could be enormous strengths in certain situations. And you pointed out anxiety and being hypervigilant at some point likely led to the survival of our ancestors.
Mm-hmm.
And people who were lackadaisical and didn't have anxiety were wiped out.
Yeah. So we have risk scores for different cancers, right? Like depending on someone's genetic profile, you're at, especially breast cancer, prostate cancer potentially, right? You can know you have a high likelihood of getting breast cancer. Therefore, you got to get a bilateral mastectomy, prophylactically, which means
sure.
Like,
well, you either have the option of that or very, very intensive surveillance, right? Or something like that. But yes, so we can identify people in the top risk category, right? And so we can stratify people into risk categories.
So then how does that translate to psychiatric disease then?
At this point, for the vast majority of them, it's not yet really a useful diagnostic or risk tool yet. Schizophrenia, for example, is probably where there's the most power. And there, if you're in the top 10 percentile of polygenic risk, you kind of have about, I think it's around a 9-fold risk for having schizophrenia. That's equal or roughly around the same risk as cannabis use during adolescence, which is about 5 to 10-fold increase risk. So maybe you could, and so the question is, where do you draw the line and how are you going to use these scores? So for example, you could screen people who are in the top 10 percentile, which is still going to be a lot of people. So how do you do that? And then what do you do with them? And how do you prevent? And so what's happening right now is people are studying prodromal schizophrenia, or schizophrenia where somebody has a major gene mutation. Like you take people with 22q deletion or some of the other few mutations, and you kind of follow them as children to see if you can identify prodromal features and maybe modify their course. With autism, again, the polygenic risk score really only explains a few percent of the kind of genetic variance. So we're not there yet. We can't really do much.
Yeah.
But I think the notion would be you want to identify people in the highest risk group and the lowest risk group. And for breast cancer, for example, just to give a concrete example, the way that would go is, maybe people in the top 10% have a 10-fold risk. And so instead of waiting till age, whatever the age is now, I'm 40 or 45 or 50 to get mammograms, you now move back 10 or 20 years earlier and you begin to screen them with ultrasound and other, and maybe MRI earlier. So you would stratify people into different screening populations based on being in high risk and low risk. The issue is that most people are going to sit in an indeterminate bucket for things like that, right? Like most people will sit at least half around the, the middle half. And for those people, it may be, until we get a lot more data, it's going to be very, very hard to make any kind of clinical recommendations or clinical judgment. So where the polygenic scores are going to work right now, I think, and be implemented will be in conditions where you can identify people who are at such high risk that it makes sense to change the screening recommendations and to intervene.
Right. Great. Now, the influence of environmental factors, I just want to hit on that very quickly. You mentioned cannabis. In my mind, a primary, big-time environmental factor that's contributing to neuropsychiatric disease, especially depression, seems to be social media, for example. And I always thought about it, and you mentioned this earlier, which it's when technology outpaces our own development.
Yes.
Right? So when the environment outpaces our own capabilities, right? So that's what I see social media being. I'm wondering what your thought is on how these environmental factors affect us to trigger these diseases from a genetic standpoint. Are these environmental factors actually changing our genetic regulation? Are they changing our genetic expression at all, or what is it?
That's a great question. I don't think that's known. The way I would look at it is somebody's brain chemistry and structure and the way it's set up predisposes them so that when there's a stress of some sort, like a social stress of like feeling left out of something, or fear, what's it, FOMO, right? But when that happens over and over again, or if somebody breaks up with them and they've had a very serious relationship, that they're just going to be more susceptible to that letdown, right? It's the way the brain is set up. So I don't think the environment is, I think the experience that's occurring is the same outward experience. It's the effect on the brain functioning that's a little bit different, and that's because of the way it's been developed and set up.
Right.
That would be my model, but it's not really known.
Yeah. Now, I want to draw your attention to traumatic brain injury.
Sure.
I think a shift in traumatic brain and in all of medicine, right, based off of this precision healthcare that you've been talking about, is to be able to identify genetic risks for CTE, chronic traumatic encephalopathy, in football players that have, or anybody that has had repetitive head injuries, right, or who's going to have a worse traumatic brain injury outcome, right? And there seems to be an underlying genetic basis to that because of the variability in TBI outcome after a head injury, right? Like someone can have a concussion, they recover in 3 days, versus someone else that has a concussion, their symptoms are a lot more severe and they don't recover for, several months. Same thing with more severe traumatic brain injury.
I think here's where precision health really could come in really nicely in large-scale medical records and large-scale populations. Let's think, genetics is one piece of it, for sure, and always has to be. And to the extent that one can pinpoint the heritability of these things, one can begin to get a sense of how much is going to be genetic versus environmental.
Mm-hmm.
So for example, you can just imagine, because this is not known, that people who have had COVID are more susceptible to repeated concussions, or dementia, or brain aging. We don't know this yet, but it's possible. And so it may be that the difference, so we don't know where the big effects are, where the money is, so to speak. It could be early viral infections. It could be chicken pox. And again, I'm just making that up. But once we have large-scale medical records, and in the medical record, we have the kind of information where you can say concussions and, where that's actually part of the medical history in large-scale populations, we'll actually be able to ask those questions and ask, what are the antecedents to people who have bad outcomes? And we'll, of course, have genetic information. So we'll be able to ask about the role of kind of genetic plus environmental. Because my sense is over the next 5 years, certainly at UCLA, we are moving to genotype as many patients as possible. We've now, in the Precision Health Biobank called Atlas, consented over around 200,000 people, have about 130 to 140,000 people who have said yes to us retaining their left-aside medical specimens, such as blood, making DNA from that and genotyping them. And we've genotyped about 65, 70,000 people already. And so in the end, you can imagine in 4 or 5 years in a place like UCLA with millions of health records, we'll have at least hundreds of thousands of people, if not 1 million people, genotyped. And if we had in the medical record measure of people's concussion, and that was, and people took good medical histories, we would, or one could add on top of that, right? One could send out through my chart, we're doing a study of concussion, please fill out this form. How many of you had, how bad have they been, right? And so you could get quantitative form, at least historical information on how people have done. And, and you could begin to ask these questions. I think that the key to really understanding something like susceptibility to TBI is going to be large-scale population-based studies on the one. On the other, study of specific populations. So one example is radiation exposure, right? Who do you study? You study people around nuclear accidents and nuclear exposures, Chernobyl. How does that, Hiroshima in terms of cancer risk from radiation, et cetera. So I know this sounds crazy, but that's why football players and other people with a lot of traumatic brain injury are being studied, right? It's because they're a special population that have been subjected, as well as soccer players and other people, hockey players we talked about a little bit, people who have, as part of the sport that they do, a very high prevalence of repeated head injury and concussion. That's going to be a special population that's going to give you certain insights, but it's not going to be the same as, it's not going to be large enough potentially to understand what the factors are in the general population. So it's a combination of those, of large-scale population studies and these kind of smaller focus studies that are going to be key.
Yeah, and I got to give props to the BU CTE Center, who just did one of the first, I think the first genome-wide association study looking at, what would predispose, or what sort of genetic profile mostly predisposes to CTE, specifically. I think it was football players, but it might have just been people with a history of repetitive head injury, and they found that ApoE4 allele significantly predisposed people to more severe CTE. So I think, we're headed in the right direction.
Exactly. And again, that fits in very interestingly with the notion that our risk for disorders really has to do with the flip side of it, our resilience. In other words, ApoE4 also is a major risk factor for Alzheimer's, which is a disorder of aging.
Right.
And it's a syndrome, it's not a single thing. And so one of the major factors is ApoE4. So that's telling us that if you have that version, the normal quote unquote insults and things that happen to you as you age affect your brain in a deleterious way more if you have that allele. So it gives you some sense of that. And in the same way, it's telling us that if you get repeated injuries and likely repeated neuroinflammation from those injuries, that the ApoE4 allele is going to be bad for you as well.
How would this look like? So in terms of drug delivery based off of someone's genetic profile?
With cancer now, about 7% of cancers have a mutation that dictates which drug would be best for that cancer. You're attacking a specific pathway and mutation there. With something like, if you want to stay with CTE, we can move off of it, but I think it's a fascinating area because it has huge implications for kind of, raising kids and kids in sports and adults in sports and kind of how we do things. And so optimally, you'd want to identify people at super high risk and protect them. How do we protect them? I don't have an answer to that. You could identify people at super low risk and give them a free pass, but now we're discriminating based on genetics, and I find that potentially troubling, right? Potentially, I'm not saying it is, but we have to understand what we're doing. So if we say you can play this sport because you have better genetics or more adaptive genetics than this other person, what is that really doing? I mean, that becomes, at some level, problematic. On the other hand, maybe that person would want to know, and maybe they would take that positively and say, thank you, you've saved my life. I'm going to have instead of playing football, I'm going to do bike racing,
Right.
or speed skating, or, or something else where I'm not going to be at risk for that. And I'd rather look at it that way,
Right.
personally.
Yeah.
I'm not saying everybody would, that we're doing this to optimize people's outcomes and to improve their lives and give them a higher quality of life. And so that is, in the end, kind of what precision health is about. Looking at the individual and helping them understand how to make the best choices for themselves.
Mm-hmm.
So we have to kind of balance autonomy versus being, the patient autonomy, and, because then it's an interesting thing. Then the child who's about to play, football is a patient almost, right, at that point, even though they don't have a problem at that point.
Right.
And so I think it's going to be a combination of kind of genetic factors. Hopefully, we'll be able to have quick, and I know a lot of people are working on this. I don't know where the field is, but about 5 or 6 years ago, I was aware of it. A lot of biomarkers that you could measure, hopefully in serum. Hopefully you don't have to do a spinal tap, but either they could be physiological, like EEG. They could be imaging, but easiest would be a blood biomarker where you could actually then assess, has somebody had a severe enough injury to be worried about? And in the end, if we could get biomarkers like that, it's probably going to be more powerful than genetic susceptibility or environmental susceptibility, because it will be specific to that insult in that patient at that time.
But that's for the diagnosis of concussion, though. I don't think that can tell you. I mean, I might be mistaken, but I don't think that would tell you, all right, you had a concussion. We can see that with GFAP, which, is one of the biomarkers. And thus, you're at risk for developing CTE. I don't think that telling you a biomarker that diagnoses concussion when already it's kind of a clinical diagnosis would tell you that would predispose
Right, so
to
there are
CTE.
different kind of biomarkers. One would be a diagnostic biomarker, another would be a prognostic biomarker, right? And they're likely to be different things.
For CTE.
For CTE, and it could be that the biomarkers have to do with the number of concussions, the level of elevation of these things, plus genetics, right? So you could see that the biomarker for developing CTE or the algorithm is going to clearly be complicated and multifactorial, but that's going to be different from knowing, did you have a concussion?
Right.
But it's potential that the severity, that maybe the genetic factors, I'm just going to make this up because I don't know, maybe the genetic factors that make an individual susceptible to having an elevated high GFAP after a concussion are the factors that predispose to CTE, and that there'll be some levels of these things that are actually representing a combination of the severity of the environmental insult,
Right.
the trauma, plus the genetics. And, so the hope is that it might be an integrator.
Right.
It might pull together all those different features. Does that make
Right, absolutely.
Yeah. And so that's not known. So maybe in mouse studies or animal studies, you could take different genetic mouse backgrounds and give them CTE. There are all kinds of things like collaborative cross, where everybody's been genotyped already, and you could just take animals from that, give them CTE, see how they vary in terms of response to these biomarkers, measure serum lipids and proteomics and metabolites, and then do kind of quantitative trait analysis to see if you can identify specific genetic variants that affect these biomarkers, and if those biomarkers relate to kind of some long-term outcomes, at least in an animal model.
Right.
That would be a start.
Right. Now let's talk about the drug delivery, because I mean that's it, right? Like certainly all this genetic profiling, a lot of the work that you're doing informs on predisposition, prognostication, diagnosis as well, but then prevention, but also treating with drugs, right? Like I mentioned before,
Yes.
treating with drugs that address that specific genetic aberration.
So somebody has a cancer mutation that causes the tumor to proliferate and invade. It's because of that mutation changes a particular pathway in the cell that makes it divide more and take over. You can target the drug, doesn't change the genetic abnormality. It's not changing it back to normal.
Mm-hmm.
It's targeting the downstream effect of that.
The molecular effect. And
Exactly, in the pathway to kind of block that.
Got it.
And so it's unlikely, the only place where that would happen with a drug would be if you use a nucleic acid drug, like a CRISPR therapeutic, to change somebody's genome. That's a different kind of thing.
Yeah.
But not recommending that for this.
Right. So just very quickly, last topic, CRISPR. Is there a space for it in precision? I don't know why I can't say that word. Precision healthcare. Is there a space for it in precision healthcare?
Absolutely. So CRISPR is the newest and most amazing form of genetic engineering that we have. It, really changed the game. There'll be others as well. It's going to continue to advance. And what that means is that with CRISPR and other things like it, we can make therapeutics that are nucleic acids, RNA or DNA, instead of small molecule drugs or proteins.
Mm-hmm.
And so I think in general, there's a huge place for that. In fact, there are numerous trials going on now with CRISPR to target eye disease, because again, the virus harboring the CRISPR construct, the nucleic acids, can be put directly into the eye tissue, easily, so it's accessible. We can do ex vivo therapy, so that means take blood stem cells out and engineer them so that they no longer have the sickle cell mutations. And Don Cohn here at UCLA is actually leading part of a trial in sickle cell to actually change that. And there's been transthyretin amyloidosis, which is a neurologic condition that we sometimes see. It can cause cardiac abnormalities or peripheral neuropathy mostly. There are 2 nucleic acid drugs on the market for that, but there's also a CRISPR therapeutic trial going on from another company, and they had preliminary good results with it.
Mm.
So I think more and more we're going to see that you'll be able to deliver CRISPR therapeutics to certain body areas that are, that are tractable.
Mm-hmm.
And then in other cases, probably take tissue out, like blood, and re-engineer it and put it back in. And the benefit of that is that it's constrained, you can check it, you can make sure there's no off target, and you can do all the quality control that you need, and then you can put it back in the patient.
Yeah.
So kind of an autologous
Right.
Treatment. And so I think there are actually numerous trials going on with that now, and I mentioned a few of the areas. So like retinitis pigmentosa, sickle cell, amyloidosis, there are a whole bunch.
Amazing.
And I think in cancer as well,
Yeah.
in terms of the CAR T-cell therapy and kind of modulating the immune system.
Yeah, it's amazing. Let's finish with neurogenetic determinism. It's something that you've talked about before, and it basically implies that, it's a topic that you can either sort of pick one opinion or another, but just the fact that your genetic profile, whatever it is that you're born with, doesn't end up determining the kind of person that you become, or potentially some of the diseases that you end up having.
If you take 2 twins who were raised apart, they'll have, identical twins. They'll have remarkable similarities, but they'll also have differences. In fact, there's twins who are twin astronauts. One spent a year up in the space station, the other one stayed down. The one, there are massive differences between those 2 environments.
Yeah.
And so we also know that environment, which are drugs. I mean, one of the things that we give, that we do with people is we change their environment by giving them drugs or giving them treatments. And we know that that can have a huge impact on people. So the genetics is just part of the equation. And, even if we were able to predict who's going to be autistic, for example, we know that environmental changes like early intervention with behavioral therapies in certain kids who are having severe social problems can have massive positive effects. What does that mean? We've actually altered the trajectory of brain development by intensive behavioral therapy. That occurs all the time.
Yeah.
So, number one, the good news is the brain is environmentally malleable. So it's not 100%, and there's a lot, but that also makes it susceptible at the same time.
Right. That's awesome. That's it.
That's it.
I know you got to go. Thanks so
Yeah.
much for coming. I mean, it's absolutely incredible work. I say it's a lot about a lot of different fields of medicine, but I feel that genetics is certainly an amazing and new forefront of medicine.
Yeah, no, it's going to change things.
Yeah.
But it'd probably be by the time you're my age.
I can't wait.
Yeah.
Is there anything you want to promote or anything like that before we sign off?
No, I'm excited about the fact that you guys have this podcast. I think it's really cool, and I think that bringing awareness to all of these really difficult topics that are hard to understand, that even we don't understand, where we're, I think it's really, it's really neat that you guys are doing this. And I was also going to say that this is a difficult area, like the, traumatic brain injury.
Yeah.
Because in the in the way that I think about things, one needs huge populations to be able to get the data that we need to use genetics and use risk scores.
Right.
And so one's going to have to be really creative about how to do that in the context of our health system.
Yeah, well, I think UCLA is the right place. It's got people like yourself and other amazing people that I think can get the job done.
I agree. That's
Awesome.
why I'm here.
Great. Thank you, everyone, for joining UCLA Brainsport Podcast. If you guys have any questions, comments, concerns, email the email below. If you guys have any questions for Dr. Geschwind, email the email below, and maybe I'll serve as the middleman, and if they have any questions, I'll forward it your way.
Sure, that would be fantastic.
Awesome. Thanks for joining. Appreciate it.
welcome.
Stay
My
safe.
pleasure.
Be well, guys.